Everyone, if you took a few minutes away, it's our really great pleasure today to welcome Dr. Steve Festin, who is presently Director of Scientific and Commercial Development at Charles River Laboratories, to talk to us about immunodeficient and humanized mouse models and how to generate them and how to use them. Dr. Festin has had a long-standing career. I'm sure it could be quite interesting to invite you not only to speak about mice, but just career-wise, how you got to where you are after doing a PhD in biochemistry and molecular biology at the Albany Medical College. Then you went on to do postdoctoral fellowship at the NCI in Maryland and became assistant professor of biology and biochemistry at Hamilton College in New York. And then you switched, basically,
Yes.
and saw the light and went on to do a number of work, 30 years of experience in scientific research and management in academic, pharmaceutical, biotechnology, and government organizations. So you've had this wonderful experience of going from both the academic to the more pharmaceutical interfacing with a number of other possibilities. And you're an expert on these mouse models that we all want to use, but are deadly afraid to buy because they're so expensive.
Yes, understood. And it's not, and they're difficult and challenging, right, to make. So, but hopefully today's talk can give a little bit of an insight into both a couple of things. One is I'll share some information about the nature of the models that we're using, as well as how we go about doing it. I'm happy to offer support, for labs that are interested in potentially going it alone to try and do this for themselves, or I think, as you'll see, especially in terms of our support of our drug discovery businesses, ready access to quality QC'd, as we would call them, or quality controlled verified animals is very important. So thank you again for the opportunity to be able to speak. As I mentioned earlier, I wish I could be there in person and be able to interact and answer questions directly. But we're all working well, I think, with the Zoom and Microsoft meetings as it may be. So just to, again, as with that transition and background, is seeing the light, or I went to the dark side, I'm not quite sure, was really going into the commercial side of the business. And so as Director of Scientific and Commercial Development for Charles River, and I'm not sure that anyone would find my career that interesting. I'm not sure that I do myself, as it's just kind of sort of worked out in a way, but I'm in a really fortunate circumstance to where I sit really between the science and the business, right? Take into account the legal aspects of it, but also, really the utility of helping to try and provide tools to the biomedical research community, identifying new technologies and helping them through the process in order to be able to make them more widely accessible, right? So the way to accelerate and move research is to have very good, useful, and current up-to-date tools in order to do that. So just, I guess, and also to give a little bit of background, and it's not intended to be all Charles River-ish here, right? But just to give a bit of a sense, I thought that the audience would appreciate a little bit about what Charles River really does. It's changed a lot in the last few years, and really where I sit within that organization, and why we've actually gone about doing what we've done, and how we've worked across our organization in sort of unique ways in order to be able to bring some of these models forward very, very quickly. And so this is this little loop-de-loop slide that we have. This is essentially just a general overview, a bit of a map of everything that Charles River actually does. Where I work in the business really sits over on the left-hand side of that in the basic research side. And Charles River, right, so that we're essentially in the research models and services division, is the original legacy Charles River that was founded by Dr. Henry Foster in 1947. So we've been around more than 70 years. years. And over that amount of time, and especially in the last decade or so, Charles River has really grown dramatically to extend into drug discovery space as well as safety assessment. And as actually many of you probably know some folks that are working at some of the Charles River sites in the area, also providing clinical support and also manufacturing. And so within that basic research is the RMS division. And so I like to refer to it as the tools business, right? So we're in the business of providing the research tools, whether it's a Black 6 mouse or it's a fully humanized CD34 NCG, we're in the business of providing research tools in order to be able to help to support whether it's drug development, basic research, or discovery. And so the research models and services groups, essentially we breed animals. Everyone I think is quite familiar with that, but we also offer other additional services. So in order to be able to breed the animals, a long time ago, laboratories were established, especially for the research animal diagnostics, which are also available. And we've also added in to where we can actually do breeding on behalf of our customers. We can bring animals to Charles River, breed them in isolators, and be able to ship and distribute them back to the client as it may be. We have embryology services, rederivation, a number of things that are really centered around producing a very high quality animal model for research. And we also share our expertise and experience through our IIS group in terms of even being able to design, build, staff, animal facilities, everything from a veterinarian to the folks that are making sure that the building runs every single day. So just a little bit of an overview and just some quick statistics and where Charles River is essentially now, and this is in 2018, still waiting on the 2019 numbers to be all wrapped up, but we're over 80 facilities in 20 countries, 85% of the drugs in 2018 in some way Charles River was involved in, and then in the research models area in particular, 1 out of 2 animal models typically produced for preclinical research globally comes from Charles River. So think about Charles River, think about being a US company, but at least a US based company, but we are essentially global, right? So we have major production sites in Europe that includes the UK, across Europe, UK, France, Germany, Italy, Charles River, Japan, and also Charles River affiliate, which is Vital River in China. And from each of those sites, the pedigree all comes from our foundations in Wilmington, but essentially reach to everywhere in the world. And in our genetically engineered models and services, which is our breeding organization, we maintain animal lines with a very large number of genetic mutations, modifications, and characteristics that we follow on a regular basis. So essentially that's where I live within Charles River. That's essentially what we do. I think most of you are probably familiar with that. And so for today, my focus, I apologize, get a little sensitive on my mouse here. Should probably click. Make it better. So for today, I'd like to talk about 4 different sections. One is just a little bit of background on immunodeficient models, using our portfolio as examples. But we're going to focus more specifically on the triple immunodeficient NCG mice. You'll learn a lot more about that, its performance as well. Talk about humanization, and I now have, I think it's 5 case studies since we introduced this since at the beginning of the year of different oncology models that are associated with the humanized mice. And you know what I should also do here is, I think the chat may disappear when I actually start showing. So if there's any questions, raise a hand, or Nicole, I can see you. So wave at me if someone has a question that we need to answer. Okay, so Charles River immunodeficient models, and this actually represents generally the classification of models that are essentially available. We break them into these 3 different categories. One are the nude models, which is essentially, it's nice because they lack hair, so subcutaneous tumors are easily measured by caliper. Others that are hair, and also we have hairless as well, and contain SCID mutation. And the 3rd classification is really these triple immunodeficients. And this is not, you'll see in in a later chart, this is somewhat progressive in terms of the genetic modifications associated with them, but essentially, especially between nudes and skids, depending on the model type, and we heard a lot of interesting comments, especially from Sebastien and Peter on PDXs as well, and being able to grow them is that very often, especially transfer from donor patients, involves implantation into multiple strains, right, to ensure the potential survival of those cells, especially those that may be a little bit less aggressive, as was earlier mentioned. Apologize. So within this classification, we have there are rats, especially in the nude. So in the nude classification, the nudes are athymic as well. We'll talk about a little bit about that, and are also on a number of backgrounds, BALB/C as well as CD1. SCID, which is a sort of severe combined immunodeficiency syndrome mutation, is also present in a number of different models. And then the triple immunodeficient, we really have just the NCG, but we have what I'll talk about today are both the PBMC humanized as well as a CD34 humanized version. So this chart I like because it kind of maps out a bit of that progression, but identifies the original genes that are really associated with the nude phenotype, both in rats and in mice. The FOXN1 is actually a spontaneous mutation that we see and is primarily associated with T-cell deficiencies, right? The SCID mutation, as I said, is a spontaneous mutation to base mutation within the PRKDC gene. The PRKDC gene is involved or associated with recombination events. And so that mutation disrupts the PRKDC function. And those animals also lack T cells and B cells. And they can also contain an additional modification, which in this case is called the hairless mutation. So there are hairless versions of this, and progressively further immunodeficient. In this case, we have the fox chase SCID beige has the PRKDC, but is also combined with the beige mutation, and also has deficient NK cell deficiencies. The most immunodeficient, as we would kind of say, is in the NCG, which lacks both T cells and B cells, but the NK cells really are unable to be activated. So I'm going to focus the call or the next part on the NCG mouse, and then I don't know if sorry, I guess it's a picture. I can share slides as well too if you need to. So the triple immunodeficient NCG mouse. So let me just give a little bit of background. So it was originally co-developed very recently. So it was brought to Charles River in 2016, but it was engineered by CRISPR using CRISPR technology in 2014 in a bit of the early stages or days. It was actually a multiplex CRISPR knockout mutation, doing indel mutation, targeting a number of genes. And this particular line was derived from that one, which had deletions in the PRKDC that had been targeted to the early and terminal stages, as well as in IL-2 receptor gamma. The background of the mouse is actually an NOD, or a non-obese diabetic mouse, which also, for a couple of reasons, it has some immunodeficient phenotypes, but also, as I'll mention and describe a little bit, has some sequence variation in the SERP alpha gene, which makes it more permissible for transplantation and transfer of human cells. So the three components of this triple immunodeficient mouse, as I mentioned, includes the NOD-SCID, and that's associated with the SERP alpha, as well as the NOD immunodeficient phenotype. The PRKDC lacks VDJ recombination, and the IL-2 receptor gamma is a common subunit of interleukin signaling. And the lack of IL-2 signaling within the NK cells makes them unable to really be activated. So the mouse is severely immunodeficient, and as a result of all the combination of these three, can also then therefore be humanized, right? So just to be clear, because humanization can be described in a number of different ways. And so in this case, the NCG model is not genetically humanized, right? It contains a number of mutations that make it immunodeficient. But by transferring a human tumor or transferring human immune cells, we're able to humanize the mouse and essentially recapitulate a system that contains both human tumors as well as human immune cells. So a number of applications, and I think this will speak well to this group, is that our focus has been, especially really more recently, really on cancer and oncology. So you'll see a number of case studies that we've done looking at, in particular, looking at checkpoint inhibitors, which are very common, especially in the drug discovery space. But these models can also be used to study immunology, since we're able to rebuild part of the immune system. It's also used for study of infectious diseases, I think for somewhat obvious reasons, but HIV being one that's most common. We do see onset of graft versus host disease in some cases, and so some of that xenogenic rejection that can occur is actually a good model for that. Oh, apologize. All right. Too sensitive. Back. Diabetes on the NOD or obese phenotype, and as well regenerative medicine and the ability to transfer some human tissues. So focusing on the three components, part of what we did and reported at AACR last year, we did exon sequencing of the SIRP-alpha gene within the NCG itself. And in this alignment, looking at the contact points, right, between key amino acids, between CD47 and SIRP-alpha, which essentially it really is associated with the recognition of, say, self versus non-self, is that we found that the NCG has significant human-like character within these sequences. So this is part of what makes it permissive for the humanization. And as well, the CRISPR targeting was specifically directed to make null mutations, right? So they were targeted to the N-terminus of each of the PRKDC and the IL-2 receptor gamma proteins using indel technologies at the time. And so those deletions disrupt the protein prior to any transmembrane section, or as well also signaling domains for each of these proteins as well. And so these are considered to be genetically null genes as a result of the CRISPR modifications in NCG. And so we have that combination, the NOD background and the PRKDC knockout and the IL-2 receptor gamma. And then especially when we initially received the animals from Nanjing University, had done some specific immunophenotyping and characterization of the NCG model. And so some basic hematology just to prove that, again, it was expected to be immunodeficient. We did a comparison between CD1, which actually has progenitor character to the NOD strain, is that we see significantly lower white blood cells overall. Lymphocytes, monocytes, and eosinophils are dramatically decreased between the NCG and the CD1. So this is essentially really what we expected to happen. We also then did immunophenotyping, looking at both blood and also at tissues, including the spleen, which I'm not showing here, but I'm happy to share with anyone if they're interested. But relative to the data listed here is relative to the CD1 strain. So this is the percent of cells that were actually identified. The means are listed here, and then the percent of the CD1, and you can see that in the peripheral blood that there are very, very low, barely detectable numbers of T cells, as well as CD4, 8, as well, and B cells here, representing very, very less than 1% of the overall immunocompetent strain, which is CD1. So we're very pleased to know that the model, again, from an immunophenotyping standpoint, was, quote, immunodeficient. And since the introduction of the model, both through our clients as well as studies that we've done internally, this is a very short list. Our list is much longer now, of a wide variety of tumor types that have actually been transferred to the NCG. And in fact, the NCG is very permissive, right? So the non-humanized NCG has been used in a number of different PDX type studies, especially engraftment of cells that are coming from PDX, and it seems to be very permissive to cell types that are derived from different tumors, sites of origin or tissues, right? And so that's actually worked out very positively, plus a number of other tumor types that grow in most other common nudes or in non-skids. In some cases, the LN cap, we actually saw better performance. Triple negative MDA, MB231s, we saw comparable performance and very good tumor growth. So there are a wide variety now of tumors that have have been demonstrated to, both in graft as well, have similar growth kinetics to, again, sort of the less immunodeficient strains, but other immunodeficient strains that are out there, triple immunodeficient strains like NSG and NOG that are currently also available and humanizable. Since that time, there's a wide variety or number of publications that have been made on the NCG that are out there, and that number continues to grow. And so it's not just Charles River who's working on this. We've seen a significant adoption of the NCG in a wide variety, a number of CROs in particular, but also academic organizations that are adopting this as well going forward. So to talk about the sort of the focus of this particular talk really is on humanization, and I'll spend most of the rest of the talk really focusing on our attempts to humanization of the NCG, and we've really looked at two different options, right? And so the first one I'm going to talk about, we refer to as CD34. So CD34 is a cell marker for stemness, and CD34 cells actually represent the hematopoietic stem cell progenitor, sort of pluripotent to both the myeloid and lymphoid lines that can be transferred to a mouse. But we also have and offer a PBMC model. And I use this slide just to really differentiate between the two because they really do have two different immunological characteristics or character. The CD34 stem cell model essentially is the most challenging model to both develop as well as to produce on a regular basis. The cells, basically it's adoptive transfer into the bone marrow. So bone marrow is ablated. We transfer CD34 cells that are derived from a number of sources, but primarily now from cord blood. And those cells then populate the bone marrow as well as the stem cell compartment and continuously both develop and differentiate and expand throughout the animal, essentially regenerating a portion of the human immune system. The benefit of this is a number of reasons, and one is that model, although it takes 3 to 4 months to actually be able to produce, but that model is also lasts for a significant amount of time afterwards, for 3 or 4 months after they've then been quality controlled. So that's a real benefit. But they also contain not just the mature cells that are listed here primarily on the bottom, but also the intermediate cells associated with hematopoiesis. So it is continuously replenishing those cells on an ongoing basis. We transfer only a few cells over, let them be able to seed, expand, differentiate, and continue to grow over time. And then at the end, I'll after we talk about the CD34, we'll talk about the peripheral blood mononuclear cell model, which is a very easy model, very quick model. It's a tail vein injection of PBMCs. The nice part about PBMCs is that they can be obtained through leukapheresis. So that's basically a patient. Oh, careful what I'm leaning on there. There we go, through leukapheresis. And so this is basically live donor cells that are then contributed, injecting those cells. Those cells are then injected, and essentially they are only mature cells. So those cells will expand. They're not really differentiating from the stem cell character. And it works out within 3 weeks to be almost exclusively a T cell model. And I'll show some data on that a little bit later. Okay. So focusing in on the CD34 stem cell humanization, talk a little bit about next. So this is really focusing the CD34, and this is on that progenitor cell adoptive transfer. So this is a bit of the promotion slide. There's some, the benefits to this, and I think basically I'll skip over this. But we've done a wide variety of validation studies, both at Charles River and with our partners, and I'll show data that's actually come out of that now over the last 9 to 12 months, and walk through the process that we use for the production of the animals. And I think it highlights a lot of the benefits of doing this. So one of the things, if you know, the laboratory is thinking about humanization, each endeavor to humanize, right? So each project then is going to take 3 to 4 months, you know, really before you're going to have sufficient numbers of cells in order to be able to set up studies. And so in looking at that, you know, having a lot of experience. And so essentially what we're doing is on a regular basis. So if I think about it now, so we're in November, right? We're really producing animals that would be available for your studies sometime in February or so, right? And so the animals that we're currently sharing were injected back over the summer, right, in July. And so we're doing that on a continuous basis, and we have a lot of experience both with the injections as well as monitoring those models as they go through. So the challenges to that are a few-fold, right? One is we have the direct colony source for the animals directly out of our notobiotics. So they're very clean, coming directly out of isolators, as opposed to maybe animal facility reared animals as well. And so these are actually, and again, is our notobiotics department is very close to the seed, right, or foundation colonies that we have for each of these colonies as well. So they're very high quality going in, and when I say that, that's both health tested, you know, as well as the animals themselves. We have a reliable donor source, so it's qualified IRB as well as otherwise, and it's coming from live births and donated cord blood. And the CD34 cells for us are actually processed. And so in your laboratory to humanize, you can actually process the cord blood yourself if you have direct access to that. For us, we have specific criteria that we work with our producers to then meet specific qualifications that we know are robust in order to be able to gain significant humanization. And so we've optimized the process. So what's the process? So on day 0, as it's shown here down at the bottom, is that we actually use X-ray irradiation, and you know, the NCG is pretty happy, you know, with about 1.6 to 2 gray for that, and it's a simple CD34 stem cell tail vein injection. So the CD34 stem cells, we actually work with frozen cells. They've been frozen down, and it's so to speak a proprietary medium, but it's essentially what all the providers are using. And those recovered cells actually do very well. And that's a real benefit for us in terms of being able to set this up and getting the timing right. Once the animals are then irradiated, we actually inject probably within a half hour to an hour, almost, you know, immediately following the X-ray treatment, and allow those cells then to circulate and also follow their markers to the stem cell compartments. Once the animals have the chance to recover, we place them into isolators. So if for those of you who might not be familiar with isolator breeding, it's essentially like a glove box. And it's the soft-sided isolators are made out of plastic. They're essentially a bubble, and that bubble is completely sealed, right? So again, everything going in is fully sterilized, and including the air all being filtered and circulated. Much of the cages and everything are already within the isolator before the animals actually are even placed into it, and also tested and verified before the animals go in. So we transfer those animals once we've completed the injections. We transfer them back to the isolator, not back to the isolator, to a new isolator. That isolator then is closed, as we would call it, right? So for the next up to 16 weeks, that isolator is going to remain closed, so no other animals are introduced to it during that time. And our health testing looks in, it matches our immunodeficient health status, which is verified, is that the animals are actually swabbed every other week. We do PCR testing on or about 6 weeks, and then at about 12 weeks we actually do a full health monitoring, right? So we submit animals to what we call our HM lab. They do a full workup on that, and animals will not be released from these isolators until they actually pass essentially the same level of health status as the original NCGs. So this process, we're very careful during the process to ensure that we limit the exposure to any possible pathogens. And then just following that at 14 weeks, we optimized it. We were doing 12 and 16-week bleeds, but 14 weeks seems to be the right spot for us to actually do the blood draws for the quality control assessment. And that data is actually, shown over here, and we actually have data out to over 36 to 37 weeks. We do see, especially during this initial period, where we have a number of B cells. We have a high level of humanization. It's maintained throughout the course of this period at somewhere, typically at 60 to 80%. Our actual minimum threshold is 25. And again, this is looking at this humanization rate is actually the ratio, right, of human CD45 versus mouse. And in looking at, we see increasing T cells over time, CD3s, CD19s tend to be fairly high early on, and then also drop off and level off during the course of the typical experiments without any problem as well. We're actually, this is very typical of what we see. And as I said, as we do these injections weekly, bi-weekly, and we follow them continuously and really are looking for meeting that initial, that highly stringent quality control, just about 14 to 16 weeks. And then once the isolator has passed the health assessment, those animals are then released for sale. So I guess that's a little bit about, talking a little bit about what's behind the scenes, right? Because for the, although fairly expensive, right, talking about the amount of work that goes into it, but from the end user standpoint is that if you are setting up studies and then reach out for to obtain animals, this week, you may have them in 2 to 4 weeks, as opposed to going through this whole 4-month process in order to produce them. And I'll also say is, we have had some, and there are some challenges, in terms of donor variability, even with the methods worked out fairly effectively. So it is expensive in part because it costs a lot for both the cells. The flow analysis, as everyone on the call probably knows, is quite expensive, and as well as the housing of the animals during that period of time. So that's a little bit about how we get to that point of actually having the originally humanized animals. The real important piece is, well, how do they work, right? And how do we go about looking at that a bit more closely? So like I said, we have 5 case studies of data that I'm showing today. I've got a few other studies that we actually are currently still analyzing some data, especially with our partners. Looking at as would be, I think it's so we've got kidney, PDX as well. We looked at a couple of different CAR T types of therapies coming forward. But these initial experiments, so these are all done. They were presented last year at the Society for Immunotherapy and Cancer in 2019. And it's collaboration between the Research Models and Services group and our discovery group in Worcester. They actually do conduct our flow analyses, but also can set up these studies. So a typical study to look at, and then these are all focused on checkpoint inhibitors, which is typically the sort of the highest demand that we have as far as clients are concerned. And it allows for the study, the interplay really between the human immune cells and the human tumor cells directly. In this case, it's A549, which is a lung cancer tumor. And it's a fairly well-growing one. So it's about 2 weeks prior to, so in the humanized animals, 2 weeks prior to actual dosing, there's inoculation of the tumors and then randomization at the beginning of the experiment. And it's typical that you'll have, there'll be a few animals that may or may not necessarily engraft to detectable levels that meet that criteria for study entry at that point, which is part of why we do the randomization as well. Dosing out and this is in days, right? So study days of 32. So this is a 1-month study. As you saw on the previous slide, we've actually tested the humanization really out for, up to once they're released at 16 weeks, we've gone out to 26 weeks as a recommendation is, to the latest to start studies, out to 32 weeks and even beyond. So there's a really nice window for being able to conduct these studies very carefully. So this is looking at, this is using an antibody specifically to PD-1. PD-1 is a checkpoint inhibitor associated with a tumor PDL1, right, tumor expressed mechanism of T cell sequestration. The anti-PD-1 antibody then interferes with that interaction and enables the activation of T cells more specifically. And as you can see in this, the left-hand graph, you're going to see a lot of graphs that look a lot like this. Is that, in the isotype controls, we see expected tumor growth, but we see this the divergence, especially in terms of the tumor growth and the kinetics, are dramatically impacted by the presence of PD-1 alone. And to show, especially in these early experiments, we saw distribution of, these are of human cells. These are the CD8, CD4 subtypes. Looking at, in these cases, these are the two different treatments. And so we don't really see dramatically different levels necessarily as a response. There's almost like the response is almost not fully detectable, or at least statistically different. But we can see that we have good presence of human immune cells in a wide variety of different tissues, especially here in the spleen, and then tumor infiltrating lymphocytes. So these are lymphocytes that actually we, so essentially at the end of the study, right, at the termination, we actually remove tissues, extract cells from those tissues, do flow analysis on them, right? And that's what you see here, is looking at the CD8, CD4 subtypes, as well as T regulatory and a number of other subtypes. But we see significant numbers of lymphocytes within those lymphocytes within those tumors themselves. So this is was a very good indication of the ability and the functionality of the humanized model. And looking back, it was our initial characterization of these models, which were ongoing, and again carried out for out to 34 to 36 weeks. So this is a little bit of a time sequential representation. So we had really done some of those studies, but also we're characterizing the humanization over time. We see sustained humanization here shown out throughout the course of this study. And in this particular, we also did flow analysis, right? So at 12 weeks, 16 weeks, we did some tissue analysis. I'll show that data. And then out to 25 weeks also did flow. And then close to the end of the study, again, just looking at the distribution of the human immune cells, as well as looking at them in the circulation. So we see CD3 over the course out to 32 weeks. Actually, as I mentioned earlier, we do see a bit of an increase and a balance of CD4 and CD8 over those same periods of time are present. And then when we looked at each of the different tissues, LWB is actually the blood, and we've got bone marrow and spleen. Not surprisingly, in the bone marrow, we actually see a bit higher in the later days. After tissue collection and accumulation, especially of these cells within the liver, and in particular, some fairly high numbers within the lung. So we have, as expected, where you would expect to see, mouse immune cells, we also see the human immune cells distributed throughout a number of the tissues. And as I mentioned a bit earlier, a few slides ago, when we were initially looking at the best time to do the quality control, we tested at 12 and 16 weeks. And so looking at CD3 T cells, B cells, as well as the CD4 and CD8 subtypes, we did still see that a bit of an increase between CD3 and the CD19. In this case, it's dropping off just a little bit, but we know that it will fall off a bit later. And then so really selected 14 weeks as being our time for bleeding and then testing, and usually we'll have the results back within a week and be able to release the animals at 16 weeks. So this is about where they would be expected to be once the animals were provided. And looking at infiltration, so this is just looking at, again, this is the two different time points of the tissue, so at day 166 and 223, looking in the bone marrow, spleen, liver, and lung, and this is the relative distribution, right, of we got CD45, as well as some of the myeloid types as well, so some accumulation within some compartments, but fairly low levels of myeloid, but we do see, again, those distribution of cells across the different tissues that are shown here. So I had already shown that the first slide really looking at checkpoint inhibitors. I probably should have had this slide a little bit before that. But essentially, there's two primary targets for checkpoint inhibition currently that actually work fairly well, but are not, have turned out not essentially really to be applicable to necessarily all patient types, as well as in particular all tumors. And so there's a lot of work that's being done looking at this basis, but also additional interventions to help to drive that. And so CTLA-4 is actually a receptor, interacts with CD80, 85 here, and involved in inactivation of T cells by antigen-presenting MHC cells. But we're really focused on, in these studies, looking at the tumor cells and these interactions that we have between the PD-1 and PD-L1, and the ability of these antibodies to then interfere. And again, that's activation of the T cells, and then the downstream elimination of the tumor by recruitment of both human and we've seen mouse cell types in order to modulate, as it would be, the tumor growth. So this slide is the same similar slide as I had provided earlier, where we saw this is the A549 lung tumor, and we see modulation of the growth and distribution in the spleen until, again, is the tumor infiltrating lymphocytes. In the spleen and in the tumor infiltrating lymphocytes, we're able to extract those cells, and then we actually treat them, right? So we activate them by basically chemical treatment, and then looking at each of the different cell types, we are also able to detect this is the fold increase in interferon gamma, IL-2, and TNF alpha. And so across the board in both the tumors and the spleen, but in particular, we see that the cells are not exhausted, that they're able to respond to stimuli and are producing factors that are known, the cytokines that are associated really with the ability to modulate the tumor growth. So very happy to see that in the A549, and you'll see in the subsequent slides and studies, we have a repeat of that. So a little bit more, so colon tumors, this is an RKO tumor, and this is using the standard of care. So Pembro is an anti-PD-1, and again, we still, again, see that rightward shift, and testing the blood, and also looking in the spleen, in the tissues, and the tumor infiltrating lymphocytes, we can see the presence of the human immune cells. CD45 is over on the left up first, is where it's expected. 4 and 8 of the T cells, Tregs and NK cells are here, as well as the B cells. And these are the human cells that are currently present, and they've all are present and have been able to infiltrate into the tumors. This is the same study, really just represented again slightly differently, right? So these are the presence of the cells in the tumor infiltrating lymphocytes. The RKO study is designed just slightly differently, and you can see is that the RKO tumor actually grows much more quickly. And so one of the things to, especially to consider in early studies, right, is to look at either in non-humanized and a background strain, or even in humanized models, is to actually look at the tumor kinetics, right? And part of what we do is we offer access to some of those animals early on for setting these studies up, so that you have some of that data, and knowledge about the engraftment and kinetics of animals prior to actually setting them up for the randomization and dosing subsequently. So the RKO, it's actually a fairly fast growing tumor, and you can see that here in how it actually really reached its endpoint fairly quickly. And we see the modulation, Keytruda, with that rightward shift in terms of the kinetics. And then looking again, this is actually looking at the T cells and their activation in the tumor infiltrating lymphocytes of the CD4 subtypes, as well as CD8 subtypes, that we see differential stimulation of those depending on if we're looking at interferon gamma, IL-2, or TNF alpha. But again, cells are responsive, and again, believe that essentially that's part of the mechanism of how they're modulating these tumors. Additional studies are done on these tumors, especially depending on the particular mechanism of action, not just of the PD-1, but we also have clients that are working with other pharmacological interventions that they can use for these kinds of studies. So one example of PDX is lung PDX. This was done in Freiburg, Germany at our oncology site there, our discovery site. This included both Pembro, which is the anti-PD-1, and ipilimumab, oh, I can never say that right. So we'll just say CTLA-4. Antibody, right? So ipilimumab, sorry. And also the combination therapy, right? We see at these doses, again, that same modulation of tumor growth in these. And this was also a somewhat unique study that it was also done in a single mouse study as well, right? And so this is looking just at the 397, but using the PDX and then a single mouse study, you're able to actually study them almost more like a human population. And this really set the model for being able to set that up because we knew that we had PDX that were responsive, and then we could look at PDX from different patients of similar types and be able to see which ones are or are not necessarily responsive. Of the the viable cells, CD45, and again, this is bone marrow and spleen are listed here. And then we're looking at tumor numbers as far as, and then under the different treatments. I think you can maybe say, looking at the CD25 Fox P3, that there may be some modulation in response to the tumor, the intervention or the pharmacological intervention in the treatment, especially in the combined therapy. And again, this was being explored further both by the client and their particular pharmacological intervention. But we do have, again, expected CD45 as well as infiltrating cells within each of the tumors. So the fourth and fifth, and again, now that we're starting to accumulate a larger number of studies, you see we're following along with a very similar study type. So this is a Colo 205. A colon cancer, and then this is actually MB 231, 436, which is also triple negative. And in each of these cases, right, so I'll focus on the colon cancer. And then this is just, again, anti-PD-1, anti-CTLA-4 in the combined therapy. We see that shift. Again, statistically significant difference between the tumor growth and the treated. And then this is, again, the tumor infiltrating lymphocytes, basically showing that those cells are permeating into the tumors, and as well, again, some data that I'm not showing here are also activatable. In the breast cancer, and again, just look at, so the study days here, this is this tumor grows sort of between the RKO and the A549 and the MB436, so inoculated around 10 days prior to the randomization, and MDA MB436 is a bit longer, a bit slower growing. You can actually kind of see that in the slope here, and a similar type of study. So these studies are all running, out within about 30 days, with both dosing and then flow analysis on both tissues as well as on samples. And so in this case, right, again, we see modulation of the growth. We see pretty significant effect of the combined therapy, but again, that actually led to significant animal loss in this particular study. So we're investigating that a little bit more closely. But it was, interesting that we did see, especially a really clearly difference, especially in TNF alpha expression in the MDA MB436. And so of the other tumor types that we've looked at by a similar means, we really haven't seen this difference or this cell subtype, right? So the CD4 type and CD8 cells is seeing, really significant differences in terms of the activation of those. And so that's one thing that we're actually looking at a bit more closely, and hopefully be able to come back and talk a bit more about next time. So the idea here is, again, I know it's a number of studies, they all look very similar, but it gives you some idea very quickly, since we've been able to do these over the last 9 to 12 months, of a wide variety of different tumor types and, ways of setting up tumor studies in a humanized mouse model to be able to study some of these interplays. So I realized as well, you know, these are all very much targeted to pharmacological types of studies, but you know, can also be used as the basis for actually, you know, understanding the mechanism of action of both these antibodies, but also what's going on between the tumors and the human immune cells in a much earlier stage of validation. So with that, I'm just going to, the last few minutes, I'm going to switch gears just over to the PBMC model. And just to remind you of the difference between the two models again. So CD34, right? So this is the stem cell transfer and then production of a reconstructed partial human immune system. And then with the PBMCs, we're really looking at mature cells, and as you'll see, almost exclusively T cells in terms of that expansion within 3 weeks post inoculation in this particular model. It's also useful, and again, you know, we were looking at checkpoint inhibitors. That applies to a number of immune cell and tumor cell interactions. But this particular option is more accessible, but it has its limitations, and so hopefully I can walk through some of those now. So in terms of the engraftment kinetics, and so Charles River, so when we first brought this forward, we set up a particular model. So we screened a number of different donors, right? And then we looked at their engraftment rates. So we tested different cell numbers for injection. We also looked at engraftment rates at CD45 over time. A very interesting observation, and I don't necessarily, you know, I don't show it here, is there is a high degree of donor variability in terms of the engraftment rates and or associated onset of GVHD. The faster that they engraft, then the earlier we see that the animals really succumb to the symptoms of GVHD, as we initially start measuring it in weight loss, but really the lack of the ability to thrive. And so we've, again, have screened a number of these and identified one that gave us a window that would allow for us to be able to study the tumor and immune cell interactions. But just, you know, just one thing to be careful or at least aware of, depending on sources of PBMCs, if they're local, like in your lab, students or others, you know, people who are around. And again, I'll admit this again. So part of through graduate school for myself, right? So I was able to keep myself sort of deep in ramen by donating my leukocytes every I think it was 6 or 8 weeks. I'd go up to the blood lab and they'd put me in one arm, out the other, and take them back. So mostly for doing platelet types of studies. But so anyway, so we again screening. So there's a very high degree of donor variability, and if the cells are engrafting very early, you see that in terms of the symptoms of GVHD. And then others, you know, they'll be very healthy and seem to be living over a very long time. But when we actually do the testing, we see that there's very low levels of expansion of cells. But we do see, you know, and our goal here was within 21 days to get about 25% of the live cells that we would see as being engrafted from the PBMCs. Studies can be run at earlier stages than this, but this is really, again, more or less what is the standard threshold for being able to initiate these studies. And we see that the PBMCs, the CD45 is sustained out. It's still continuing to expand and is sustained out well, actually past day 41. So the tumor kinetics are here, or I'm sorry, the engraftment kinetics basically show for this particular donor, which we call our PBMC Select, that we offer with the NCG kit, that we see very good reproducibility. So an interesting, those observations, so with there's high donor variability, but that donor and then repeat donations, right? We do see consistency really in terms of the engraftments for those, which is interesting as well. So the idea here was to try and identify a window, and that we can see that the majority, as I said earlier, almost all of those cells of the CD45 cells are eventually T cells, and we see actually a fairly good balance between the helpers and cytotoxic T T cells across the study lifetime and study windows. And then this is a graph looking at body weight versus the days. And as I mentioned earlier, part of what we were trying to do, and again, so strongly recommend if you're setting these studies up, is to screen a number of donors, look at different cell numbers. And so you can identify, and I just showed you the PBMC expansion and the CD45 cells. And we were targeting this study window where we have survival of the animals. And in this case, actually, through the course of the full study, we actually had 60% of them actually make it, right? So 3 out of the 5, really didn't suffer too significantly, but within this study window of up to 45 days. So we had about 20 days out past 45 days of 3 to 4 weeks for being able to look at the tumor interplay. And then we, as I mentioned earlier, is that we really do see as a result, as we measure body weight, but there are other indicators clearly of the onset of GVHD at the later stages here of these studies. Having optimized that and then looking at a similar type of study that we've done before, this is actually with the RKO. This is with the RKO tumor line, and this is in the presence on the right with PBMCs and in the absence of the PBMCs. And again, ipilimumab, got it, and the CTLA-4 and PD-1 inhibitors, you really don't see a dramatic pharmacological effect, but in the presence of the PBMCs during the same study type of window. And if you look at the study window, it's about 25 days. Here too, is that you've got tumor growth and as well are able to modulate this. The only, again, I would say the challenges here are twofold. One is identifying donors that give you that a good profile in order to be able to set up these studies. And number two, you need to consider this as being a system, right? So in setting it up and running through the studies, it's likely to be set up as a system to be able to go and to look at a number of different things and being able to generate these animals on a regular basis. So strongly recommend sort of optimization and tweaking this, to ensure that you've got the right window for the tumors that you're working with. Very, very important. So just a brief summary in comparison, and this is again all based on our experience and then with the NCG model, is that so the PBMC has a couple of real advantages in that it's rapid generation, right? So you have the animals and then the cells are frozen. We bring them out, warm them up, count them, do tail vein injections, and we can do tumor engraftments, a week later so that they're growing, enough to be within that window, and somewhat easy standardization. Part of the disadvantages, are it's transient. So these are mature cells. You don't have hematopoietic development going on. They're localized primarily in the peritoneum, but also you see that these cells are primarily mature types. It's good for short-term studies, setting up a study, screening, being able to look more closely at specific interactions between T cells, as I mentioned, and tumors. And the advantages of the CD34 are somewhat obvious and straightforward, right? So it's a bit more robust, but has a broad spectrum, right, of human hematopoiesis. So a number of different cell types. It's suitable for larger cohort generation. And we're doing that by lots. We actually do single donor injections and for production of each lot. And so you're able to actually develop your study if it's you would want to work from different donors to be able to combine them, or even to be able to screen different studies starting with different donor cohorts. As I said, they're thoroughly standard for the kinetics of the reconstitution. We literally are doing this every week or so, and it has been highly reproducible in terms of the ability to produce these. We've only had, a couple of experiments that did not quite go as well, but otherwise we've been very successful in achieving the number of quality controlled animals we needed to get to. So some of the disadvantages are, so the T cell selection is really in the murine thymus. This is actually part of how it avoids the GVHD, which is actually, somewhat of a benefit. But as a result, right, is actually really, based on the mouse MHC to some extent. It's good for long-term studies, as I said. So this can go out, even say 2 months, right? Maybe 3 months might be a bit of a stretch, but that's significantly longer than the 2 to 3 or 4 weeks that you might might have with the PBMC, and can be used for a number of these studies. So these studies for efficacy, toxicity studies are actually currently going on in Laval in our safety assessment site, specifically on the NCG background, and we'll have data on that coming next year, and looking at specifically at cancer therapy. So I just get to talk about it and all of the other things in terms of bringing it together. This is actually the poster that we presented on that has the A549 data on it, as well as the characterization initially presented last year. On the left is Jenny Rowe. So she is our humanization scientist. So she actually came out of a humanization lab at Upstate Medical Center in Syracuse, and then worked for a company that was running humanized studies for a number of years before coming to Charles River. So we're really fortunate to have her. She is the lead scientist in producing these animals, and literally, you know, she's hands-on in producing these on a weekly basis. And then from our Worcester site in Discovery, on the right-hand side is Christoph Eberle. He's enjoying himself at SITSI, well-deserved. He is our flow scientist, right? So he lives, eats, and breathes, and comes actually originally from a human CLIA immuno testing laboratory in Germany. And so just to give credit to where credit is due, and my contact information is here, and you have it on it shared through the site. If you feel that you have any questions, we're happy to offer any guidance or input, you know, whether it's on our background strain or otherwise. I know this is a very Charles River presentation, but I think that a lot of what we're talking, you know, what I've talked about is applicable to a number of different situations and settings. So with that, I will stop, and I think we have some time to take some questions on the last section.
Thank you so much. This was great. I really enjoyed it. I have two questions already. So in the PBMC model, do you try to select donors that are HLA matched to the tumor, or in your case, it doesn't matter all that much?
Yeah, so that's an additional advantage, especially of the PBMC model, right? So we're limited, right, with the CD34 coming from cord blood, right? We don't have repeat donors. You know, it's an N of 1 for each situation. So one of the advantages of the PBMC model is, yes, you can look at haplotype and other factors, especially, and even work with disease cells, right? So, you know, one of the things more recently looking at, say, even COVID positive patients, right, and otherwise in terms of the performance of those kinds of cells. So it does, the PBMC does offer that advantage. For checkpoint inhibition studies, typically the HLA is not a factor, or at least for us, it hasn't been a significant factor, especially in modulating the PD-1 studies that we're doing. So those are designed intentionally really to try and necessarily avoid that a bit. But the PBMC does allow you that flexibility, right, to be able to do that. And, you know, Charles River now does have resources of multiple donor sets of PBMCs, you know, that can be accessible, can be potentially screened and then used for studies. So it does. As I said, the CD34 model is much more difficult given the amount of time, right, and also the random nature, especially of you know, the source of the cells that are coming in, and they're also, you know, again, those cells are actually maturing really within the mouse organs and infrastructure.
My second question is for the other model, the CD34 humanized mouse model. Given the fact that there's so much now development and interaction between microbiome and the immune system, and a lot of that has to do with tumor development, has anyone checked whether humanizing with the CD34 actually changes anything in the microbiome of these mice?
So, so we haven't done that definitive study, right? And that's actually another, you know, especially particular focus area for us in terms of looking at that. The part of the challenge is, and again, but I would actually expect that, right? Just in general, just, you know, things like stress and otherwise, right? You know, really do, you know, lead to significant changes in terms of the microbiome where they can. And so we haven't necessarily done that definitive study, but I'm just going to, you know, offer some free advice though, right? Is that especially to run microbiome studies, because we do have germ-free Black 6 and otherwise, right? The housing conditions essentially for being able to maintain, even if you're maintaining animals that are not germ-free, is that the protocol should be essentially at the level of germ-free, just essentially to ensure that you have stability within the microbiome within whatever locations they may be, or even your different study groups, right? Very difficult, and it's pretty, it's interesting. I'm done model creation for a long period of time, and I'm a molecular biologist, and it must be about the genome, but you have to turn the clock back 70 years to and again, even 50 years ago they were producing germ-free animals, and how they're housed and bred and maintained actually become the key to running really successful microbiome studies. Having the tools to be able to study it like next-gen sequencing, and then being able to look at it a bit more closely. So no, we haven't done that, but again, this is sets up a bit of an ideal situation to be able to do that. We just haven't had the time yet to really be able to set them up that way.
Any other questions? No questions at all. Peter? No? Well, Steve, your presentation was recorded. We have your coordinates. It's been great having you. We are very thrilled that you were able to have the waves cross the country. And we hope to be able in the future to bring you in person in Montreal to discuss more at length, and to see the further development of these models, and how we can best use them for the variety of experiments that our people are doing. As you saw this afternoon, some of the presentations but there's like a whole set of other people doing a whole bunch of other things, and I'm sure that going further, they would be thrilled to meet with you. So
I'd love to hear more about them too, the different kinds of studies and applications. So yes. Especially, beyond the focus that we've had. So yeah, that would be great.
So this will certainly do this again for sure. So thank you very much. And it's been very nice to see all the development that your company has done. And I'm sure it's also been great probably to put it on the map for all of those trainees who might not necessarily know about Charles River. They're starting off, they're introduced slowly and surely to the new developments in these fields, and it's great to be able to see that many different companies are actually working very intensely to develop the tools that make their experiments worthwhile and significant. Thank you so much. So this concludes our workshop for today. We will be sending everyone a short survey just to get a feel of what you think about this, and whether you have some suggestions to improve the further delivery of other workshops or further topics that you'd like to hear. We will also include in that message the email addresses of all of our speakers today. So if you need to get in touch with them for more information, or be in contact for collaborations, or maybe decide to do a PhD somewhere else, then this is certainly a possibility. So thank you very much, everyone, for being there. We hit at one point we hit 70 participants today, so we're very pleased about that. So thank you, everyone. Bye-bye. Thanks.